Hi, welcome to this video on Lowry assay for protein quantification. This is a commonly used method in any biochemistry, cell biology or molecular biology lab. This was published in 1951 by Lowry et al. In Journal of Biological Chemistry. Therefore, this is a gold standard method. Therefore, I will discuss it in detail so that you understand the components or the requirements and their role in this method. In addition, I will also discuss the underlying basic principle, how to set up the assay and a standard curve using bovine serum albumin or BSA. Its calibration, precautions, sensitivity and limitations will also be discussed in this video. So let us begin. Then the first question is why to measure protein concentration in a given sample or biological sample. As you know, proteins are important biomolecules because they perform biological functions, regulate metabolic reactions, physiology, traits, and our phenotypes. And aberrant expression of certain proteins have been associated with the disease pathogenesis. Therefore, it is very important to understand or to measure the concentration of protein in a given sample for understanding disease diagnosis and pathogenesis. So the next question is what types of protein assays are available? Broadly, you can classify these assays into two categories. The first one is the non-selective or general methods. It includes three I have listed here. The first one is you can measure protein concentration by measuring optical density at 280 nanometer directly, and this is because of the properties of the property of aromatic amino acid in the protein. However, this method is not sensitive and therefore it is not used much. The other methods listed here are Lowry method, which I am going to discuss today, and the third one is Bradford. These two methods are very commonly used. These are gold standard. Today I will present Lowry method, and on some other day I will present Bradford method as well, but later on. So these are non-selective or general methods. They are not protein for a particular protein. They will measure all kind of proteins. The other group of tests which are used are included in this, and we can say that these are specific or protein selective test, and this include Western blot analysis or ECL, Western blot analysis, dot blot, immunoprecipitation, or ELISA. You can use any of these. They have their own limitations and the merits and demerits, but these are all based on immunological assays and utilizes the specific antibodies. So both methods basically are complementary to each other. Specific measurement, specific technique can only be used if you know the protein concentration, general protein concentration or total protein concentration in a biological sample. So just to give you overview point of Lowry method or Lowry assay, it is a non-selective gold standard. I say gold standard because commonly used, routinely used in any lab, in almost every lab. It is a colorimetric test method in which the color change is measured as an indicator of the level of protein in the sample. So a colorimetric method which uses a chromogen, and chromogen here is Folin reagent that changes the color on the reaction with protein. So the 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 intensity of the color is proportional to the amount of protein present in the sample. So principles underlying this assay. In this assay, in the first step, protein sample is treated with copper sulfate, alkaline copper sulfate solution for 30 minutes. In the protein, I have shown here 3 amino acids, tyrosine, tryptophan, and cysteine. They play a key role in this test. That is why I have shown here. In the first step, a copper protein complex is formed. In the second step, this is treated, the copper protein complex is treated with Folin reagent, which is a strong oxidizing agent, and these amino acid side chains basically reduces the Folin reagent to a blue color chromogen. This color intensity is directly proportional to the amount of protein present in the sample. Therefore, it is quantitative, and this intensity is read at 750 nanometer using a spectrophotometer or colorimeter. So let us discuss how the copper protein complex is formed in step 1. Here I have shown 4 protein for sake of simplicity or convenience. However, in biological samples you may have several different type of proteins. So copper basically here makes coordination bond with the nitrogen of the backbone or peptide bond, which is shown here with the red color. So these 4 proteins, they make coordination bond with copper. This the tetravalent is the most common, but in some cases you will see 5 coordination bond. In other cases you might see 6, but 5 and 6 are less common. Most common is tetravalent. So this is how the complex is formed, and let us see in second step how this protein complex, copper protein complex, reduces the Folin reagent. So how this protein, copper protein complex formed in step 1 reduces the Folin reagent. In Folin reagent, you have phosphomolybdenum tungstate, which is reduced to heteropolymolybdenum, which is blue color chromogen, and this color intensity is read at 750 nanometer using colorimeter or spectrophotometer. Now this reduction reaction, the 3 amino acid side chain participate in this reaction, and these amino acid I have shown earlier are tyrosine, tryptophan, and cysteine. Now, in order to understand the mechanism in detail, you have to know the requirements, the components used in this. So, what are the requirements? First is you have to have a copper sulfate solution in alkaline medium. Second, you have to have Folin-Ciocalteu reagent or Folin reagent. These are the name of two scientists, Folin and Ciocalteu. And the protein samples. It may be purified protein, it may be cell or tissue lysate. I have been using these method commonly, the protein sample, and the success of your experimentation also depend to a larger extent how good is your protein samples prepared. So I will discuss this also issue, and I hope that will help you. And the last one is spectrophotometer. You should know what is the basis of spectrophotometer, and if you don't, please read about Lambert's and Beer's and Lambert's law. That will help you to understand this. So let us see the first requirement, copper reagent. It is copper sulfate 1% in alkaline medium, pH is 10, and this is basically sodium carbonate buffer. Sodium carbonate in 1 molar NaOH, pH is around 10, and it also has sodium potassium tartrate. It helps to keep the copper sulfate in soluble state. So, and sodium carbonate provide alkaline pH, and copper sulfate provide copper for the formation of copper protein complex. Now this is very important that you mix these, prepare these and mix just before use in a ratio of 1:98:1 in ml of A, B, and C written here. So mix these A, B, C in the ratio given here, and it is ready for your assay. So let us come to the second requirement or the component, which is Folin reagent. Folin-Ciocalteu reagent, this is also called. So it has phosphomolybdic acid or phosphotungstate 10% each to keep this in soluble state. These are actually the strong oxidizing agent, and they oxidizes the protein basically complex. And lithium sulfate is stabilizing agent. It keeps the solubility well of these two component here, and phosphoric acid gives the pH close to 1. So this is also diluted. You 1:1 before use, 1:1 with distilled water, and You can buy these components from the company, but you should know what are the function of these components. So if you have any problem, you can sort out. So the third requirement is cell or tissue lysate. It is very important to prepare the cell lysate or tissue lysate very well. Now for that, you take a tube and put about 0.5 gram of tissue, freshly resected tissue. Avoid using frozen as much as you can, and add MOPS buffer PS7, 5 ml of this. It should be chilled. Everything should be kept on ice. Now in the next step, you chop the tissue with scissors finely and polytron to release the protein into the medium. So you obtain crude lysate in this tube, and in the next step you spin it at 800 G to remove cell debris and collect the supernatant by passing through cheesecloth, and you collect here this one. So this is like supernatant or crude lysate. So from this tube number 3, you further clarify by removing mitochondria and nucleus by spinning it at 10,000 G for 10 minutes, 4 degrees centigrade, and collect the supernatant in the new tube. This is called tissue lysate or crude lysate, and this will be used for the measurement of total protein in the sample. So this is the final sample tissue lysate or crude lysate, whatever you call. You measure the total protein content in this tissue lysate using Lowry method. We are going to discuss. Adjust the protein concentration ranging from 1 to 3 milligram per ml depending on the concentration in your tissue lysate here. You can use MOPS buffer PS7 for this purpose also, and you aliquot the lysate and store at minus 80 degrees centigrade. Avoid frequent freezing and thawing for better result. So setting up the reaction, you label two tubes. One is, this is the sample. You add protein 2 microliter from your sample and add 1 ml copper reagent in the tube. In the second tube, don't add protein or any sample here. You add just copper reagent 1 ml. Incubate for 30 minutes at room temperature. Add Folin reagent. Incubate another 30 minutes and you will see color change. The content, you will see color change in the sample tube but not in the blank. You transfer this into cuvette and measure optical density at 750 nanometer using blank, which you prepared here, in a colorimeter which is shown here. So you will get optical density. What does this mean? How you will translate it into microgram? And for that you have to have a standard curve, which we are going to prepare in the next slide. To set up the standard using BSA, label 9 tubes 0 to 8 and add BSA. In the first tube, this is blank, don't add any BSA, it says 0. In the remaining tube, you add a different concentration of microgram as shown here. Then add 1 ml of copper reagent, incubate at room temperature for 30 minutes. Then add Folin reagent in the tubes. Additional incubation for additional 30 minutes and you will see color change. In the blank there is no color, but in the remaining tube you will see color concentration dependent intensity increase in the color. So you transfer the content or the solution into the cuvette and measure optical density at 750 nanometer using blank you prepared here, and you use these values to prepare a standard curve between optical density and BSA concentration. So standard curve calibration, we have obtained the optical density for different concentrations of BSA and these are the values shown by these stars. You connect these point and you will get a straight line curve.
So this is called a standard curve. Using this, you can measure the concentration in your sample. So in this case, for example, your optical density of your sample is here 1.6 or 1.7. You can extrapolate this and you will get this value of protein concentration equal to 6 microgram per ml in this case. So using standard curve, you can calculate, you convert the optical density into microgram per ml. So this is how you convert optical density of your sample into microgram. So there are certain precautions for the success of your experiment. You must observe these. The first one is the reagent must be freshly prepared or diluted. You can buy from the companies if you want. There are many companies, commercial companies, they are selling the whole kit, and follow the instructions, and that will be good enough. Run sample in duplicate and triplicate or triplicate. This will minimize the errors, a standard error. And you have to prepare a standard curve using different concentration of BSA simultaneously together with the protein sample, and also together with a blank. Blank is a background which is subtracted from each sample while you are reading at 750 nanometer.
So I have discussed all goodies about the Lowry assay, but there are certain problems also, although they are of minor nature. For example, there may be interference from the chemicals present in a sample, such as glycerol, carbohydrate, or certain type of detergent. They basically inhibit the formation of copper complex in the reaction. There are certain cations, divalent cation, particularly calcium and magnesium, or chelators of cations such as EDTA and EGTA. They basically affect the formation of copper protein complex also. EDTA and EGTA are commonly used as protease inhibitor while you are preparing your protein samples. In addition, beta-mercaptoethanol and DTT, they are commonly used as protease inhibitors. They also reduce Folin reagent independent of protein, and therefore they will give an false overestimation of protein in the sample. However, there are certain chemicals such as SDS, which is known to circumvent the effect of interference. So regarding the advantage, it is fairly accurate and extremely sensitive technique which can measure from 0.5 to 1 microgram per ml concentration in a given sample. Therefore, it is a gold standard method. It is suitable for dilute samples such as cell lysate or tissue extract or enzyme preparation. However, it is labor intensive and time consuming technique. In the end, I would like to thank you for watching this video, and I urge you to like it, share it, and subscribe it. Until next time, you take care and bye bye.